he is editor of many journals. He has published several papers. He is author of several patents also. Without any delay, Dr. Rai, please, we are, waiting for your presentation. Please go ahead and start, Dr. Rai. Thank you.
Okay, thanks, Dr. Ashish Thaplia, for this wonderful introduction. And I still think that I am a student as well, a learner as well, a researcher as well, like you all people. So with this note, welcome to all and good afternoon to all of you. And you all are the undergraduate student in microbiology and biotechnology program. So I'm trying to take your say 40-45 minutes, maybe 1 hour, but we'll discuss the basic concepts, basic ideas about microbiology, and I'm trying to give you an idea about the virus, immunity, and vaccines. So let me confirm from my wonderful audience, am I audible to you? Am I clear? And is the slide is visible to you?
Yes, sir. Yes,
Okay,
sir. Yes,
okay,
sir.
thanks a lot.
Yes,
Thanks.
sir.
Good, fine. So now you can see the first slide. Here I'm showing the title virus, immunity and vaccine. I've actually missed one word as well because there is a correlation between virus, immunity, vaccines and the nutrition. So in the coming slide, you will realize that how the microorganisms, immunity, nutrition, vaccines, all are closely related or interrelated. So the first slide, if you see, this picture is depicting the representation of one of the virus, right? And we are trying to say that, okay, the viruses are deadly, are pathogenic, are harmful, and causes infection and disease. In the second picture, if you see, this is showing a man, a person who is actually defending the entry of microorganisms. And this picture is showing the defense mechanism in the body, how the body defense mechanism work, and how our body recognizes self and non-self. And the third picture is showing vaccines, right? A representation of vaccines, syringes, and the vials. So with this, I welcome all my lovely students of microbiology and biotechnology. Uh, so moving ahead. Okay, so during my this topic, during my this interaction with you people, and I would like to listen from you students as well. We're going to discuss about the microbial diversity.
Dr. Saab, sorry
Then,
to cut you off, Dr. Saab.
yeah, sir.
We would be grateful, if you could just talk, 2 minutes about what your research is and what you have done, so that students also know, and if they are interested, then they can talk to you.
Okay, fine. Right, so uh, I am Dr. Nishant Rai, and actually I did my MSc in microbiology, right? And during my postgraduate dissertation program, I was working on cattle virus, right? And trying to clone and characterize the gene of bovine herpes virus. And since I was very much interested in virology, so I did my PhD in the area of rabies virus. And I actually worked on DNA vaccines against rabies virus under a project that was ICR project known as National Agriculture Technology Project, NATP project, VVT project. So I completed my PhD in microbiology, and my area of work was virology, some kind of immunology, cell culture techniques, etc. during last 2 decades, I'm working in the area of biological characterization of herbal plants. We can say uh, um, a drug which is of a natural origin, which is from herbal origin, right? And against diseases including candidiasis, which is caused by candida. So our team is also working in area of different plants, including green tea, and we did lots of work in the area of green tea, and we have also published and filed patent where we have identified the active constituents present in green tea as an antimicrobial agent. So, if you are interested to do any kind of work in the laboratory, it is very important to go in two-phase work. The very first is to understand the basic concepts of instruments or instrumentation. Actually, that will help you to do a work or to optimally utilize the instrument for your work along with the troubleshooting. And the second part is to understand the problem, to identify the problem. And the problem is, for example, if you say corona, yeah, it's a problem which is caused by virus, and how you can uh, control for the societal benefits. So you will find a problem and you will find a solution for the benefit of the human welfare. So with this, so I'm trying to give you a brief about the concept. So I'm trying to give you an idea about this microbial diversity, a little bit about viruses, virions, morphology, composition, then little bit about features, properties, and body defense mechanism, how it works, and vaccines, right? So we'll take up the pictures and we'll take up the notes from the councils or health organizations as well. Moving ahead, yeah. So if you see this slide, you will find that I have shown a picture from on the left hand side. You will find different type of designs, right? And you will find some circular structures, small, little bit large, and then you will find some irregular structures, right? And here you will find some different type of margins. So it is actually the colony morphology of a bacteria. On the other hand, if you see this picture, you will find that the bacteria grown on a plate showing a growth as a form of colonies, and there are different type of colors observed, different margins, different shapes, different labels, different colors you will identify. Likewise, the same picture is also depicting different type of structures of organisms grown on a suitable medium. And right hand side, in the bottom, you will find different shapes of viruses, right? The circular shapes, the rod shaped, like filamental shaped. So this all is giving you an idea that microorganisms are highly diverse. And if I say diverse, it means it shows diversity in all sense. So can you confirm, is it visible to you? The whiteboard I am showing you, is it visible to you?
Yes, sir.
Yes,
Yes,
sir.
Uh,
sir.
Yes.
right. So when I'm saying microbial diversity, it is very interesting to know, because many people, many researchers, many scientists quoted the unseen world of microorganisms, the unseen world of microbes. This statement is telling that the microorganisms are so many that it is a complete world which has not been explored completely yet. And the microorganisms, when I'm saying microorganisms, I believe that you can understand what does it mean. It means all the bacteria, yeast, fungi, viruses, etc. They are highly diverse. And this diversity is on the basis of its shape. यानी कि microorganisms are diverse because they show different shapes. They shows different metabolic properties or metabolisms. They shows diversity in their habitat or ecology. And when I'm saying habitat or ecology, it means the conditions where the microorganisms reside or live. It includes the extreme conditions as well. So if you think, okay, can microorganisms survive at a very high temperature? The answer is yes. Whether it can thrive at very low temperature? Answer is yes. High salt concentration? Yes. So it also shows diversity in causing diseases. We call it as a pathogenicity. Disease causing ability. It shows diversity in their genome as well, right? So their genome also shows variations, means it may be same. So microorganisms, am I audible?
Yes,
Yes,
sir.
sir.
Yeah, okay, good. So microorganisms shows diversity in different aspects. They shows diversity in their morphology, that is shape. You will find different type of shape of microorganisms. Their metabolic pathways are different. They follow different cycle to get the nutrition. The basic nutritional requirement of microorganisms vary. Likewise, their habitat varies, their pathogenicity and disease causing ability varies, their genome varies, and hence the immunogenicity. Immunogenicity, yani the genome or the genes which are present in the microbes also shows immunogenicity and it varies. And to it, in cut short, I can say the microorganisms are highly diverse in nature. And this is the beauty of this microorganisms that encourage us to do research, to do findings, because these microorganisms has two faces. Two faces. One is the dark face and another is the bright face. When I'm saying dark face, it means it is the face that causes diseases, infections, right? And we call these microorganisms are harmful or pathogenic. These are termed as pathogens. I request you mute yourself, kindly mute yourself so that I can. Yeah, thank you. Thanks a lot. So what I'm telling you, there are two phases of microorganism. One is the dark phase, another is the bright phase. Dark phase, that is, they are harmful, they are pathogenic. Means they have the ability to cause disease. And the bright phase is that it is beneficial for us, right? They are beneficial for us. Means microorganisms reside in our body and maintains the physiology of our body. So in many terms, they are beneficial because they are also used in fermentation of food, or we call it as a fermented food. So this picture, this slide is depicting the diversity of microorganisms, and it also gives an indication. Okay, it means the microorganisms shows diversity in their genome, evolve and cause infections and cause diseases. And this is why we regularly explore different microorganisms which are emerging microbial diseases, microorganisms. Moving ahead. So a student asks, okay, what is virus? And we have lots of discussion. You know that, okay, virus is not a cellular, right? It is a cellular molecule. So virus, which is basically a acellular molecule, which is called as nucleocapsid molecule. And when I'm saying nucleocapsid molecule, means this virus is composed of nucleic acid and capsid protein. And this nucleic acid, it is actually covered by a protein coat which is called as capsid. If you see the structure of a bacteria, you will find a layer of peptidoglycan present in the cell wall, and it gives rigidity, it gives shape, it gives a particular arrangement to a microorganisms, to a bacteria. Likewise, here also, if you see that the virus is composed of only two things, nucleic acid and capsid protein. So it means this nucleic acid is enveloped by or covered by a protein coat, which is basically composed of a unit capsomers. These capsomers arrange themselves to give a definite shape to the viruses. यानी कि ऐसा समझते हैं कि इस न्यूक्लिक एसिड के चारों तरफ पाया जाने वाला ये जो यूनिट है, सब यूनिट है, कैप्सोमर्स या प्रोटोमर्स, ये इस तरीके से अरेंज हो रहा है विच इज गिविंग अ वायरस अ डेफिनेट शेप, राइट? So virus is basically a nucleic acid and capsid protein, but still this nucleic acid, it may be either DNA or RNA. But if you go for the diversity in nucleic acid, you will find that, okay, this nucleic acid may be DNA, maybe RNA, it may be double stranded, it may be single stranded. And the single stranded molecule, it may be fragmented. It may be positive sense or negative sense. It may have reverse transcriptase. So my summation is this nucleic acid shows diversity in viruses. It varies actually. And it is actually responsible for the characteristic property of feature of a virus, means the pathogenicity, the disease causing ability of a virus is due to its diversity in genome. So if you see this picture, you will find this viral genome, it is covered by a capsid protein, and you will find the different shapes of the viruses. Like here, this is a icosahedral structure, which is called as polyhedral. Poly matlab many, और हेड्रल मतलब फेसेस, सेल यानी कई सर हैं इसके, कई हेड्स हैं इसकी. Polyhedral, it may be helical, it may be a tadpole-like structure. So if I want to draw a simple picture of a virus, I will say, okay, this virus is a nucleocapsid molecule and it is actually the nucleic acid and covered by a nucleic acid covered by a capsid molecule. That's it. But some viruses are also exposed which has extra layer. We call it as an envelope outside this capsid protein. We call it as an enveloped viruses. And these envelope holds different surface proteins. आपने बहुत सारे ऐसे स्ट्रक्चर्स देखे होंगे जहां पर आपने इस तरह से लॉलीपॉप लाइक स्ट्रक्चर देखा होगा. We call it as a spike or papillomas, right? And these proteins varies in their nature because this has different functions. For example, HA protein, which is responsible for hemagglutination of RBCs, right? Or neuraminidase protein. So you will find different proteins present on the surface which has a different functions. So this may be envelope protein, viruses, and this envelope, it can be carbo, it can be lipid, it can be protein in nature. It varies, again varies. So it means, okay, the envelope is also very different viruses. But one more word we have used to say is virion. 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 Can any one of you tell me what is virion? A virus, a cellular, a nucleocapsid molecule. But
Sir,
what
a
do
complete virus.
a complete virus in what sense?
When
Because
it contains a nucleic acid as well as a protein.
okay, so if it is containing nucleic acid and capsid protein, we are calling it as a virus. Then what is the difference between virus and virion?
Sir, virion can be a
Sir, may
single
I?
virus particle.
Okay, yeah, please go ahead. Yes,
Sir,
yes.
virion is the active infectious form of the virus.
Absolutely right. Absolutely right. So what she is trying to say is she is saying that it is an active infectious virus particle, and that guy was saying it is a complete. So I am putting it here also, complete active infectious virus particle. So this statement is giving you an idea. Okay, is it maybe in a form of inactive? राइट क्या इसका मतलब वायरस एक इनएक्टिव फॉर्म में भी हो सकता है when it is inside the body. I'm talking about when it is inside the body. So if a virus is inside the body, it is in an active form, means it is in infectious form. If it means it has an ability to cause infection, it is virion. But it may go into the inactive form and then it called as a virus because it also shows latent stage or latent infection, right? Yeah, so moving ahead. So this is giving you an idea, the structural organization of a virus. Yeah, so before going to this part, let me give you a brief about the virus properties and features. Because when I'm saying it is a acellular, but it is intracellular parasite, right? It is a acellular, it is an intracellular parasite. It is small, and when I'm saying small, it means it is about 30 to 300 nanometer. So it means it is smaller than bacteria. It is small and you can see under the electron microscope, but you can see its effect, its growth under the inverted microscope. It contains DNA or RNA, right? And it requires intracellular parasite, it requires a living system for its growth. Now the point is how these microorganisms are able to enter into the body. That is the portal of entry. What are the different points where this viruses can enter or the microorganisms can enter into the body, because it is a portal of entry. So depending upon its mode of infection, it enters into the body by different ways, right? So it means the microorganisms or microbes enter into the body, right, through different routes like eyes, nose, mouth, right? And when we say nose, it means it is a inhalatory route or respiratory route. Likewise, if it is mouth, it means it's a oral route, right? Different reasons are actually supports the entry of microorganisms like contaminated hand, contaminated food, etc. actually favors the entry of the microorganisms inside the body. And when it enters inside the body, it shows pathogenicity. Right? So it means it start causing infection. It follows its route of pathogenicity. But on the other hand, the body defense mechanism, we call it as a host defense mechanism, also work. And this body host defense mechanism works against this pathogen. Right? So it means there is a direct relation of microorganisms which is causing pathogenicity, the body defense mechanism, and this body defense mechanism is supported by our body immunity. Immunity, I need to resist the infection, and this immunity is supported by the nutrition what we are getting. So this concept works. Yeah, I am putting this first. Yes. Yeah, so here this slide is showing the different methods for cultivation of viruses. One of the feature of a virus that it is intracellular parasite. So it requires a living system for its growth. And you can grow viruses, you can cultivate viruses, you can even detect viruses, you can prepare vaccines using these methods. The very first method is very common is a inoculation of a virus in an embryonated egg. You can see this picture where a person or researcher is trying to inject a virus in a fertile egg, right? So what happens? We procure the fertile egg, we screen the egg for the presence of the embryo, and for this we have a torch, or we have some closed chambers with a light system where we can see the embryo, and the color of the embryo shows or indicates the conditions. If the color of the embryo is pink, it means it is live, it is healthy. And if it is yellow, it means it is contaminated or dead. But if it is darken in color, like dark green, brown, black color, it means it is contaminated or infected, because the shell may get damaged and allows the entry of other uh, contaminating microorganisms. So what we do first, we procure the fertile egg, we screen the egg for the presence of embryo, then we label it, and after this we identify the route of inoculation. And for your kind information, we actually determine the dose of the virus, right? It is called as lethal dose. In case of embryo or this is fertile egg, it is called as embryonated infectious dose, EID, and it is calculated by Reed and Munch method. Just to understand the things, you बस इतना समझिए कि viruses are virus dose are determined, and it is called as LD50 or EID50 or TCID50. So suitable dose of virus is injected. But for this injection, it is hard to inject the syringe directly into the shell. It is difficult because it will crack. So what we do, we actually identify the egg, and let me show this by a picture, a very quick picture. Yeah, okay. So let me make a, yeah, so this is embryo, this is a yolk sac, this is chorioallantoic membrane, there is amnion, Okay, then you will find air sac. So you will find air sac here somewhere, and the outer layer is the shell, followed by a shell membrane. This is yolk sac. It is for the nourishment of the embryo, growing embryo. This is chorioallantoic membrane, this part, and this chorioallantoic cavity. It actually contains the discarded part of the embryo. It is connected, right? And this embryo is covered by a cushion. Fine. Now, what we'll do, we'll identify the specific site for inoculation of virus. So I'm not going in detail. So okay, what are the factors that decide which route of inoculation or which site of inoculation is to be done, right? It depends on so many things. But for your kind information, what we do, we make a mark using this torch. Let us suppose we want to inoculate at this point. So we'll mark here, we'll make a mark here and make a hole, right? And this hole is done by a special tool we call it a driller, and this driller make a small hole, right? And we use a special syringe, right? Special syringes are used, we call it the tuberculin syringes of a particular dia and particular length, and is used to inject. So from this point, we inject the virus at this particular site. After this injection, then we seal this hole using paraffin wax or tapes, right? And then we put this egg at 37 degrees centigrade at 62% humidity in a egg incubator, right? So it allows the virus to multiply at a particular site. So this picture is depicting this part, okay, how we can inoculate the virus. The second is inoculation of virus into animals. Yeah, so we also screen animals, small animals, as per the guidelines of CPCSCA. I mean, there is a guideline. There is a committee actually which decide or which gives a clear-cut guidelines for handling of viruses, prevention, and any kind of research. So we select small animals, including mice, and there are different type of inoculation routes like intracerebral, subcutaneous, intraperitoneal, intranasal. So again, these different routes of inoculation depends upon the purpose of your study. For example, if you want to study the effect of a neurotropic virus, आप चाहते हैं कि neurotropic virus का effect क्या होगा, you will inject through intracerebral route. So there are different routes of virus inoculation. And obviously the health of the animal, the strain of the animal, right? Then genetic evolution of the animal, different aspects are first considered before inoculating any virus. And we'll identify the different changes in the animals. Likewise, the third one, very important, tissue culture technique or cell culture technique. This is very common, right? Because this is used for widened application, cell culture technique, because in case of virology, it is also used for the cultivation of the viruses, to identify the growth of the viruses, to even maintain or preserve the viruses, as well as to prepare vaccines for the viruses, right? This picture is showing, actually this picture is a picture of a cell lines. Let me tell you that when we isolate an organ from an animal, right, then the organ is made up of, is broken down into tissue, and tissue is broken down to cells, right? So what we do, we actually grow the cells, we cultivate the cells in the laboratory in a suitable medium. We call it as a growth medium. And when you grow in the laboratory in an incubator, you will see the growth of the cells, and the cells will grow uniformly as a monolayer, and it shows a lawn structure, lawn-like. We call it as a confluent growth. But when you infect the virus, you will find clear zones or areas, right? And you will observe the different changes in the morphology of the cells, and we called it as a cytopathic effect or CPE. So for information, these methods are very commonly used for the cultivation of the viruses in the laboratory. This is for your information. So till this time we have discussed about the viruses, right? I have skipped the properties and features of viruses, but trying to say you that okay, the microorganisms which are pathogenic in nature, right? It has ability to cause disease, and what happens when they enters into the body. So when the enters into the body by different routes of entry, the body defense mechanism works, and this table is showing the body defense mechanism. So obviously the body defense mechanism works in different ways. One is non-specific and another is specific. So non-specific is not specific to a particular microorganism. It is applicable to all. So like for example, your skin, which act as a mechanical barrier. So it inhibit the entry of microorganisms inside the body, right? Different secretions of the body, even the complement proteins. These are some kind of proteins present in the body, and it involved in three biological processes like inflammation, cytolysis, opsonization. So in short, what we can say is, okay, the body defense mechanism work in two ways. One is non-specific and another is specific. So what happens when a specific microorganisms enter into the body? So our body recognizes that microorganisms because it is foreign, and it responds. And in that case, it gives two types of immunity. One is called as humoral immunity and another is cell-mediated immunity. But no point is a pathogen or infecting organism is entering in our body system, and the body duality, the immune duality is working. It is either producing antibodies or it is producing some kind of T cells. And this concept is also going to be used for vaccine preparation, right? So it is actually the body's natural defense mechanism. So if you see this immunity part, acquired immunity, active, it comes under this part, artificial acquired active immunity, we call it as a vaccines. But if I say acquired passive immunity, artificial acquired passive immunity, it comes the antiserum or the antibodies. So you can inject either vaccines or antibodies inside the body. So what is happening when the microorganism is entering the body? It is actually triggering, it is inducing the body defense mechanism. And on the other hand, it is also causing pathogenicity, is causing disease in our body depending upon the severity of the infection. And this immunity is directly correlated with your nutrition. So how you can strengthen your immunity? Through food and nutrition, right? And you come to know, okay, the food, what we consume has different health benefits, including the fermented foods, which are providing the beneficial microorganisms to our body. So the body defense mechanism can be increased by this nutrition. So it is actually relating the body immunity. Moving ahead. So coming to this point, I have taken this picture or the slide from the WHO site and talking about the vaccines. Now the idea is, okay, the virus is pathogenic. Virus is pathogenic. It has an ability to cause disease. Let us suppose, right? So if I know about the detail of this virus, I can understand, okay, this virus has some part which is actually the immunogenic in nature. Means this virus as a whole or its part can trigger the body defense mechanism. Means the virus as a whole or its part can induce body defense mechanism or immunity condition. It should not be pathogenic, right? It should not be pathogenic, means it should be weakened enough that it has no ability to cause disease, and we call it as an attenuation. So if you see the WHO site, you will find the definition of a vaccine. It is from WHO side. A vaccine, which is a tiny, in a very simple language, a vaccine is a tiny, weakened, non-dangerous fragment of the organism. It's part of the organism, which includes the part of antigen, right? So it means it has some part, what we are calling it is as antigen, as a foreign substance, which has an ability to induce the body immune response. Means it has enough that our body can learn to build a specific antibody. And then if the body encounters the real antigen later as part of a real organism, it already knows how to defeat it. So what it is saying that if you know the virus or the part of the virus which has a property to induce immune response, you can use it as a vaccines. And that's why we make vaccines in different ways. So this is showing a very beautiful diagram. Here what is showing the existing pathogen, existing antibody, because the body knows this organism and this antigen is recognized by our body defense mechanism, B cells or T cells. Likewise, flu, we have an antibody. For example, chickenpox, we have antibody. But if it is a new pathogen means either the pathogen has evolved, it's a new emerging, or it has mutated. यानी कि उस पैथोजन ने अपने आप को म्यूटेट कर लिया है, अपने चेंज कर लिया है, उस पे पाए जाने वाले जो ये जो एंटीजेंस हैं वो अब रिकॉग्नाइज नहीं हो रहे हैं, देन देयर इज नो एंटीबॉडी and it is able to cause infections. So that's why the body defense mechanism work against that part. So if you visit the CDC site, that is Center for Disease Control and Prevention, you will find different aspects of these vaccines. So what are vaccines, how they are prepared, what are the concepts behind preparing the vaccines. So taking this, I've chosen this slide from WHO, how vaccines are developed. And if you open this slide, you will find this part, that this vaccine is actually a mixture of antigen. We'll see what is antigen here, adjuvant, preservatives, stabilizers, surfactants, residuals, and diluents. So what is antigen here? So antigen is actually the part of the organism. It may be a peptide, it may be a protein, or even you can take a genome of that virus, means a DNA, that is a DNA vaccine. So antigen can be anything. You will take preservatives, we take stabilizers, we take surfactants, diluents, and the most important thing is adjuvants. The adjuvant which actually enhance the body immune response. How? Because when you inject this vaccine inside the body, this adjuvant releases it slowly, and thus our body produces the immune response against that antigen, what we're calling it a vaccine. So adjuvant has a role to increase the body immune response in terms of more antibody production inside the body for a longer time, for a whole life. And it is achieved by a booster dose, including this adjuvant. And about the vaccine development stage, there are different phases: phase 1, phase 2, phase 3, phase 4. And actually it depends upon different parameters like the number of people, yeah, their age conditions, health conditions, etc. So these are the different phases uh, of vaccine preparation. So I'm not going in detail, but still I would like to share video with you people. Yeah, so this I would like to share. Please confirm, is it audible to you or not? Okay, so what I'm doing, I am sharing it again and putting my computer sound on and again sharing it. And please confirm, is it visible, is it audible and visible to you or not?
Developing a vaccine is a long road full of checkpoints. Different
Is it audible?
Yes, sir.
phases of testing are designed to make sure it is both effective and safe. But with the whole world struggling to contain a pandemic and unprecedented amounts of scientific work taking place, the road to finding the COVID-19 vaccine looks more like this. But first, let's take a look at the usual path. During preclinical trials, the vaccine is tested on animals to determine whether they develop antibodies to the disease-causing germ. If these trials are successful, the candidate vaccine moves on to phase 1 trials in humans, during which the vaccine is given to a small number of healthy adults to assess its safety in humans. The results of every phase are reviewed by independent clinical experts who assess the research team's findings. Sometimes they'll pause the trials to check the results, always making sure the candidate vaccine is safe. The next phase involves testing a larger group of people to find out the appropriate dose, whether it stimulates an immune response, and of course, to assess its safety. Once the results have been checked by the independent expert observers, the candidate vaccine is evaluated among thousands more volunteers. This phase determines whether it is effective against disease. If the answer is yes, the vaccine is independently assessed by regulators for effectiveness, safety, and manufacturing quality. It is also assessed by independent policymakers who determine the best way to use the vaccine. If the assessment meets the regulatory standards, the product can be authorized for use, and the vaccine is then ready for licensing and registration. Once that happens, the approved vaccine can be used in the population. But because of the urgent need for a COVID-19 vaccine, some of the phases can run in parallel, and the research team can submit a request for temporary authorization from national regulatory authorities and an emergency use listing from WHO. This authorization and WHO emergency listing are only given if the vaccine meets strict safety and effectiveness criteria. But the road doesn't stop there. Once the vaccine is in use, it continues to be monitored by health authorities for safety and for effectiveness to ensure the vaccine maintains the highest standards. The race is on to find vaccines for COVID-19, which is why some phases are happening at the same time. But no one is cutting corners. Everyone is subject to the same checks and independent monitoring. So although we'll get the COVID-19 vaccines faster, they'll be as safe as other approved vaccines.
Yeah, so now I would like to share this video as well. We call it as an emergency use listing vaccines. In the current scenario,
Making vaccines, tests and medicines available can take a long time, even when there's a health emergency like COVID-19. To ensure rapid access for all, WHO has developed the EUL Emergency Use Listing. This video explains how EUL works. Before vaccines, medicines and diagnostic tests can reach people, they must be evaluated to make sure their quality, safety and effectiveness are up to standard. This protects people from potential harm and ensures the products do what they are meant to do: prevent, test and treat disease. But many countries don't have the resources to carry out this crucial step, which can cause delays in getting life-saving products to those who most need them. The challenge is even harder when a disease is new, like with COVID-19. New COVID-19 health products must be checked thoroughly, yet quickly, to ensure they're effective and safe for use. To speed up this process, WHO has set up Emergency Use Listing, EUL, which assesses the suitability of health products on a risk versus benefit basis. For tests, we assess the available quality, safety and performance data with independent experts. Several COVID-19 tests have already complied with our requirements and been listed for use. For vaccines and medicines, we assess the data from clinical trials and other data on safety, effectiveness and quality, and invite experts from national health authorities to review them too. Once a vaccine, medicine or test has been listed for emergency use, we work with our partners and experts to explain its benefits and help with the approval process in different countries. But we don't stop there. WHO will keep doing regular quality checks of all products. The goal? To support countries around the globe to deliver high-quality, safe and effective vaccines, tests and medicines as quickly and safely as possible.
Okay, so thanks for watching this video. And the very last, which is very informative to all of you, I request you to visit the site where you will find FAQs about the vaccines and immunization. Like, what is vaccination? Why is vaccination important? How does a vaccine work? How do vaccines protect individuals and communities, including herd immunity? Why should I get vaccinated? What diseases do vaccines prevent? When should I get vaccinated or vaccinate my child? So we'll get many FAQs at WHO site. You will find the concept of vaccination. Being a student of microbiology and biotechnology, I would say that both part, the microorganisms, to understand the microorganisms means the problem statement about the disease causing ability of the microorganisms or emerging microorganisms on one way, on one hand, and how to make pre-exposure prophylaxis, that is vaccines, is very important. So if you know the immunogenic part of the organism, right, you can use it to prepare vaccine. For example, if you are trying to prepare a DNA vaccines, you can take immunogenic gene from that pathogenic organism or same kind of organism and clone in a suitable expression vector. Once it is cloned, it is used to inject inside the body, right, from animals starting, and you can assess the efficacy, stability of that product, right? So a biotechnologist can prepare a vaccines against a virus or any of the pathogen using genes as well. And nowadays RNA or DNA based vaccines are also emerging, right, because of very fewer side effects or no side effects or no cold chain requirements, etc. So with this note, the take home message is the microorganisms on one hand, then body defense mechanism, and the vaccines all are interrelated. If you know about the microorganisms, when I'm saying you know, means you know the morphology, structure, genome, pathogenicity of the microorganisms. You can understand how to treat that microorganisms. You know the complete genome of that organism. And if you know the pattern of its infection or pathogenicity in our body, you can able to create a vaccine which can protect you from that pathogen for a longer period or for a longer time. So this concept of understanding is new to you from the point of view you are an undergraduate student of first semester, but is interesting and you can explore different areas of work, different research in this particular direction, including the cell culture techniques for the identification, detection, vaccination, preparation of against the pathogen. So with this, I'm ending this lecture with a note that still to learn so many things from microorganisms, and there are still many microorganisms which are coming in future as well, because trillions, millions of microorganisms are still not explored and will come as one of the emerging microorganisms. With this, thank you to all of you, and would like to interact with you people. If you have any query, if you have any suggestion, if you have any comments, if you want to say anything, you are most welcome.
Yeah, thank you, Dr. Rai. Thank you very much. And now we are open for the questions. Any questions from anyone? Please go ahead and ask.
Sir, I have one question.
Yes, yes.
So you have
So
to
how do we
mention. Please, you have to mention your name and the program, and then you go ahead with the question.
Okay, sir. So my name is Shruti Joshi from BSc Honor Biotechnology. And so my question is that, so how do we weaken the virus for making the vaccines?
Okay, Shruti, from biotechnology student, a very good question and very important question. Basically, in immunology, we learn to prepare vaccines, and in that case, we use the word attenuation, where we call it as a weakening the pathogen. For this purpose, there are different methods of attenuating a pathogen, including this virus. One method is by chemical treatment. Second method is by giving passage. Passage is a term which is used to subculture a virus from one living system to another. For example, if you are using an embryonated egg for the cultivation of a virus or for the vaccine purpose, and if you transfer it into the another egg, we call it as a passage. So above 40 passage, you can able to weaken, and we call it as a embryonated egg passage vaccine. So temperature, chemicals, passage, different methods are used to weaken the virus. We call it as a weakened virus, inactivated virus, we call it as a killed virus. But still, many researchers have explored the possibility of reversal of that pathogen as well, either due to some changes in the microorganism. So reversal chances are also there, but is more effective comparatively. So you will find different detailed protocol of this attenuation. Shruti, I hope that you can
Okay.
understand and will learn more about this thing.
Yes, I got it, sir. Thank you, sir.
Welcome, Shruti. Yeah, if you have any doubt, if you have any query, if you have any comment, if you want to add something, if you want to share any knowledge about this topic, you are most welcome, because I'm trying to make it interactive and hoping that all of you will interact with this using this
Any further
method.
questions?
Excuse me, sir.
Yes.
Good afternoon, sir.
Good
My
afternoon.
name is Pratibha Rani from BSc Honors Biotech.
Okay, Pratibha.
And sir, my question is, we say that the viruses are host specific. So
Okay.
is there any other factor except the viral receptors which make the virus host specific?
Okay, so again, very good question, and it actually comes from the point diversity. When we are saying diversity, we also discuss the host or its occurrence. We use the word occurrence, viral occurrence or host system. So it is very clear that the virus structural organization, or we can say its genome expression or its requirement for expression varies, and on broader way we say plant virus, animal bacterial virus, human virus, veterinary virus, cyanobacteria, cyanophages, right, different. And if we narrow down also, they are genera or species specific as well. So it is found that the receptors varies among the host, and their metabolic cycle or its pathogenic cycle also varies, because their entry, right, it also varies among the viruses. So the people study, for example, if you narrow down the pathogen, this your bacterial viruses, bacteriophages, you will surprised to know that the bacterial viruses also varies in different genera. For example, this your E. coli phase, because it is a coli phase, infect E. coli, right? And it infects Escherichia system only because it recognizes the receptors present in the E. coli, which is lipopolysaccharide. Likewise, Salmonella Shigella, even all are gram-negative bacteria, but it varies. So its binding ability, its binding affinity, its the relation of the virus and the host varies in different system, and also varies on the basis of its genome